Hi there, my name is Drew Terasaki-Hart. I'm a PhD candidate at UC Berkeley, and today I'm going to give you a very quick tutorial on Geonomics, a new Python package that my colleagues and I put out to simulate landscape genomics. While this will be a really quick and dirty tutorial, I'm sure you'll have a lot of leftover questions. Please keep an eye out for our upcoming paper in molecular biology and evolution, which will go into a lot more detail and include validations, tests, and examples. And also consider downloading the source code and data for this notebook to follow along, which you can get at this GitHub repository here. I'll start with a brief introduction as a conceptual framework of genomics. Genomics models are essentially agent-based models composed of two major pieces, a landscape and the species. Species consist of numerous individuals, and those individuals are described by characteristics including their sex, their age, their XY coordinate positions, and their genetics. They can have both neutral genotypes across an arbitrary length genome and genotypes for one or more traits underlain by one or more loci. Landscapes consist of numerous landscape layers. Here you can see two of them, and the layers can influence the behavior of the model through three different mechanisms. One, they can serve as variables that drive natural selection on a given trait. Two, they can control the density or carrying capacity across the landscape. And three, they can influence the movement of individuals through the landscape. Movement is the first of four phases in each of the discrete time steps in the genomics models, followed by mating with dispersal of offspring, mortality, which is both density dependent and driven by natural selection, and optionally environmental change, which takes place as a stepwise event across model time, allowing for non-stationary environmental change. To get started with genomics, we'd recommend that you head over to the documentation at geonomics.readthedocs.org. When you land there, you'll see our beautiful logo, some getting started information, detailed information about how the model works, and perhaps most importantly, a parameters guide with a detailed paragraph for every single parameter in the model explaining how it works and how you can use it. Installation for genomics is very straightforward. You can use pip install genomics to install your package, like you would for many Python packages. And once it is installed, you can then begin to use it. To use the package, you'll start by importing genomics as GNX canonically. This is what's used in the documentation. And then you can create a model. The simplest, quickest way of doing that is by calling run default model. And what you'll see is that you get a model that goes through a burn-in phase, which continues until numerically and spatially the population becomes stable. And then genomes are assigned to individuals. And then the main phase begins, which for the default model runs for 25 time steps, printing out population size, births and deaths, and other information at each time step, and plotting your population of individuals as black dots on top of the landscape layer, which in this case is a blue to red layer scaled 0 to 1. However, genomics can do much more complex modeling than that, and we're going to take a look at a complex model using Sasquatch. I chose Sasquatch. I couldn't help myself because some of you may have seen that mythical creature was a taxonomic category available for talks in evolution, and I needed to use it. So I got in touch with Dr. Jeff Lozier at the University of Alabama, who put out a fun little paper years ago about the potential pitfalls of ecological niche modeling by modeling the niche of the Sasquatch across Washington, Oregon, and California. He sent along that niche model. Thankfully, he was willing to humor me, and I paired that model with the bioclimatic variables that were the most important predictors of the model, Bio19 and Bio8. And I created a nested set of directories containing the starting layers for our three environmental variables and the layers that they would change to under future climate change coming out of a CMIP model, and marked by the time steps in our model at which the environmental change will happen. I then ran the code that you see here to generate a genomics parameters file, which I can scroll through here. And you will see that this is a long and complicated file. There's a lot in it, but this is all auto-generated by Geonomics based on the call to the code above. And the user only needs to open this file, find the parameters of interest, edit their values as needed. And then with this fully edited parameters file, you can input the file to the make model function. It'll take a few minutes to run for a complex model like this. It has to generate some data structures up front that optimize runtime during the time steps downstream. But once you've done that, you will wind up with a model object, in this case called mod, and then you can begin to run it. Or in this case, walk it. The walk method allows us to run it for a fixed number of time steps, stopping intermediately, to inspect the state of the model. This is very helpful for interactive use of geonomics. And you can see that I've run this, I've given it 10,000 time steps. It'll use less than that for the burn-in. And then it'll plot a pair of plots showing the phenotypes for our Sasquatches for traits 0 and 1. And you can see that this burn-in ran for about 189 time steps, at which point it finished. The genomes were assigned to individuals, and we have our starting population with their genomes with no local adaptation yet because they have not undergone natural selection, but that happens in the next 40 time steps, during which time local adaptation builds up as a pattern of background matching between the individual Sasquatches and their local environments. And then we can see that during the climate change portion of our model, the environmental layers begin to change out every 5 time steps, and the net result is a shift in our environmental gradients with a shift in the pattern of local adaptation of our Sasquatches matching that. So this should hopefully give you a sense for the sort of modeling that genomics is capable of doing. Thank you for your time, and thank you to everyone listed here for the enormous amount of help and support they provided over the past years developing the software. And I would encourage you to please get in touch. If you're a new or hopeful user, if you've got questions, suggestions, or bugs, I'm more than happy to chat, to help you get set up, and to troubleshoot problems as they arise. You can reach me at drew.hart@berkeley.edu. And again, you can find our documentation online.